Bismillah ar-Rahman ar-Rahim. Assalamu alaikum, dear students. This is my first time giving you this online lecture. Inshallah, it will be clear for you. I'll try to go slow, so bear with me. And it might be a bit long, this lecture. So again, you can stop at any time, relax, drink coffee, and come back to me. So let's start with the introduction. Introduction of the biochemistry of hormones, definitions, synthesis, control of synthesis, and so on. So we'll talk about that. Let's start with defining what endocrine, the word endocrine means. So basically, the endocrine system is a collection of ductless glands. They secrete these chemical messengers called hormones. We'll talk about hormones in a second. Now, some tissues can also secrete hormones, like kidneys, liver, and heart, except that they're not called, they're not part of the endocrine gland. So the word endocrine means internal secretion of biologically active substances, and this is in contrast to the word exocrine, which is exemplified by sweat glands, where secretion occurs outside the body. So, what does the word hormone mean? Well, it comes from the Greek word hormao, and it means I excite or arouse. And they are basically substances, chemicals, that are released by endocrine glands and maybe some other tissues. They must be transported via the bloodstream to tissues, so that they're called hormones. And they have specific functions. They have certain effects on cells. There are about 50 known hormones that exist in our body, and we are discovering and identifying more of these hormones with the Human Genome Project and further discovery initiatives. This is a video that talks about the endocrine, that talk about hormone molecules, so you can watch it at your home. So let's go on. In order for hormones to be effective, they have to bind to receptors. They cannot act by themselves, except for one of them, which is nitric oxide. It can be considered as a hormone, but we'll talk more about it in the CNS module in the third year. So we will focus on the majority of hormones that bind to receptors. And hormones can be classified into two types according to the receptors that they bind to. Hormones are lipid-soluble or water-soluble. Now, lipid-soluble hormones, what they do is that they can diffuse through the plasma membrane into the cell. They bind to a receptor, and then the receptor hormone complex binds to DNA regions, and they act as transcription factors. Now, the other class of hormones, which are the water-soluble hormones, what they do is that they're secreted by the glandular cell. They diffuse into the bloodstream, and then they reach their destination, the target cell, where they bind to a receptor that is present on the cell surface, transmitting a signal into the cell, and finally you have an action. So there are two types of hormones, or depending on hormone classes, depending on the type of receptor that they bind to. Now, there is a major challenge. The challenge here is that the amount of hormone that exists in the bloodstream is really very minimal. It's about it's about atto to nanomolar ranges, and this is really little compared to other substances that exist in the blood, that exist in the micro to millimolar range. So the idea here is that you have very little amount of hormone that must exert a strong action, an effective action in the body. So in order for that to happen, binding of the hormone to the receptor must be specific. It must be saturable as well. That is, you have a certain number of receptors that can bind to hormone level. If the hormone level goes up, not a lot of action takes place. So, and usually the concentration of of hormones that exist in the bloodstream is about 20 folds more than the dissociation constant. So what is the dissociation constant? Well, if you look at this formula right here, where you have hormone plus receptor, they bind to a certain affinity and they can dissociate as well. The dissociation constant is equal to the amount of the free molecules, whether we're talking about hormone or receptor, over the concentration of the hormone receptor complex. Usually the KD values are about 10 to the minus 9 to 10 to the minus 11 molar, and usually the concentration of hormones is 20 times more, 20 folds or 20 times more than the dissociation constant. Now, the binding of the hormone to the receptor on the cell surface, for example, is very specific. So there is no, there's little inaccuracy or imprecision in binding. All right. There's something, so one of the things about biochemistry and endocrine gland and homeostasis and these biochemical reactions is that exists in the body. As we talked about before in the molecular biology course or the biochemistry course, an important aspect is regulation. And you will notice in this lecture that there's a huge amount of regulation that takes place in the endocrine system. One level of regulation is that the location of glands in our body is not really random. It's not haphazard. Rather, they're organized in a certain way for a certain reason. And that is, maybe one reason is to prevent the dilution factor or the dilution effect of hormones in the body. So for example, in spermatogenesis, it occurs in the seminiferous tubules, which are next to the Leydig cells. Now, the Leydig cells produce the testosterone, which is required for spermatogenesis. Similarly, glucose, the level of glucose in the blood is regulated by the liver, which is under the control of two hormones, the insulin and the glucagon. And both of these hormones are produced in the pancreas. And this pancreas is in close proximity to the liver. So again, there is no dilution, there's little dilution of these two hormones in the blood. Hypothalamus and the anterior pituitary gland are also close to each other, so that the regulatory hormones released by the hypothalamus can reach the pituitary gland nearby at high concentrations. Finally, the adrenal cortex produces cortisol, and this cortisol is required for the adrenal medulla in order to synthesize catecholamines, and they are again next to each other as well. All right. Now, a fascinating aspect of the endocrine system is that it works in conjunction with the nervous system. So you would have the nervous system, for example, affecting and regulating the endocrine system, and vice versa as well. So you have the central nervous system affecting the hypothalamus. Hypothalamus produces a number of hormones, like the thyroid releasing factor or gonadotropin releasing factor, and so on. And these different hormones can affect the anterior pituitary, producing more hormones, like the follicle stimulating factor, luteinizing hormone, follicle stimulating hormone, luteinizing hormone, and so on, and eventually affecting different organs. And what happens is that the hormones that are produced by these organs can regulate by feedback mechanism. They can regulate the release of the hormones from the anterior pituitary gland. And also these hormones the hormones released by the anterior pituitary gland can also feedback, regulate the release of hormones from the hypothalamus. So there's this, conjunction or association between these two systems working together. And in fact, some of these are some differences and similarities between the nervous system and the endocrine system, so please look at them and read them carefully. Now, in fact, if we define hormones, we can define them, or we can classify them into two types broadly. we have true hormones which are secreted by the endocrine gland, and these include the thyroxine and epinephrine and estradiol, etc. But we also have neurohormones, which are these chemicals that are secreted by the nerve cells, and they can affect other organs like muscles, for example. And some of these chemicals, some of these hormones can also act like neurotransmitters as well. So it depends on differences in terms of from which cell they are released, which cell they affect, where are they secreted, or where they are secreted into. If they are secreted in the vicinity, then we're talking about neurotransmitters. If we're talking about secretion of these molecules into the bloodstream, then we can consider them as hormones. So there are differences between these. It's the same molecule but different actions depending on the target. And these are some of the molecules that can function both as hormones or as neurotransmitters. So, for example, you look at the first one, the endorphins. Endorphins can function as neurotransmitters where they bind to pain receptors in the brain. But if they are released from the hypothalamus into the blood, affecting different organs, then we can consider the endorphins as hormones. And there are different types like estrogen, progesterone, testosterone, epinephrine, norepinephrine, and so on. So again, you can look at these hormones. Now, I want you to know the names of these hormones more than knowing what they do exactly, because you will learn about what they do in different sections of this module or other modules. All right. So, how do hormones function? Now, the type of response is different according to the hormone and the target cell. So, communication between cells using hormones can be classified or categorized into different types. So we have different types of responses. So hormones can function via an endocrine mechanism. This is a case where a hormone is released into the bloodstream. So it travels a long distance, relatively, affecting a target organ that is far away from the hormone-producing organ. Well, some of these hormones can also function in a paracrine mechanism. This is whereby you have two different cell types, one of them producing the hormone and the other one is the target cell. But they are in the same region, in the same vicinity. Now, hormones can also function in an autocrine fashion, meaning that you have release of a hormone by a cell, and this hormone affects the same cell that produces the hormone, or it can affect a nearby cell, but of the same type. This is an autocrine mechanism. We have a neuroendocrine mechanism as well, whereby you have communication between nerve cells and the distant target cells. Now, there are other types of responses that are less common, let's say, in terms of the commonly used definition of hormones. We have the intracrine mechanism, whereby the hormone acts within the cell itself, so it doesn't get released outside of the cell. We have a juxtacrine mechanism, so the hormone is actually bound to the cell surface and it can interact with a neighboring cell. And we have a pheromonal mechanism, which is something that is commonly found in yeast, and it controls sensation and responses in a different organism rather than a cell. So, do humans have pheromones? Yes, they do. So you have a person producing a certain pheromone that gives a certain smell. So hopefully everyone's taking a shower, and it can affect actually the behavior of the other organism or the other human being in front of that person. And this is why perfumes are famous and widely used. So, now, again, it's really important to regulate hormones, and they are regulated at different levels. They can be regulated at the level of synthesis, release, transport, metabolism, delivery to the surface or interior of the target cell, and finally intracellular signaling. We will touch upon these different levels during this lecture and the next lectures as well, the next 2 lectures as well. So, but there is a concept that we need to discuss first, and that is the target cell concept. Now, the target cell is the one whereby the hormone binds to, or the ligand can bind to. Now, we have 2 different types of differentiated cells in the human body. We have about 75 trillion cells, maybe more than that. And each one of them can actually respond to a hormone, which is amazing. So you can have, there's a lot of diversity in the endocrine system, and it has to be regulated very efficiently and meticulously. You can have one hormone that can bind or affect different cell types. We have a certain cell that can be affected by several hormones producing different behaviors. And you can have one hormone having several effects depending on the cell type that it binds to. And that depends on the receptor that it binds to, but it also depends mainly on the intracellular molecules that transmit this signal and other co-regulators. So we have factors that affect the concentration of hormones at the target cell. And these factors include the rate of synthesis and secretion of the hormone. So synthesis can be fast or slow. Release can be fast or slow. And the amount of released hormone is actually an important factor. Also, how close the target cell is to the cell that produces the hormone. So this is a dilution factor. So if the target cell is far away from the hormone producing cell, then you would expect that the amount of the effect would be diluted, since not a whole lot of hormone would reach the target cell. The dissociation constant is also important. That is the affinity of the hormone to the receptor. Then we have the rate of converting the inactive hormone to the fully active form of that hormone. This is really an important factor as well. So you can have an inactive hormone, but if the enzyme or the process that converts this inactive hormone to the active form is deficient, then it's useless in that case. And finally, we have the rate of clearance from plasma. All right. And we have also factors that affect the response of the target cell. That includes the number, activity, and occupancy of receptors. So you have a limited number of receptors that exist on the cell surface. So depending on how much hormone there is, the affinity of hormone towards the receptor, and saturation level as well, that can determine response. And then we have the metabolism of the hormone in the target cell. So if we have a hormone that goes into the cell, well, what would happen to that hormone? Would it be modified or not? Like the steroid hormones, for example. How do they get modified inside the cell? There are other factors within the target cell that can determine the response rate as well. So we have secondary messengers that we will talk about, like cyclic AMP, cyclic GMP, for example, calcium level, calcium ions. Well, these factors are also important in determining the level of response or the intensity of the response by the target cell. And finally, we have also the regulation of the number of receptors on the cell surface, because some receptors, when they bind to the hormone, they get internalized, and some of them get degraded. So the concept of desensitization is important. If you have large amount of hormone produced, secreted, and bound to the receptor, you have a desensitization. You have internalization of the receptor into the cell so that there wouldn't be any more receptor on the cell surface. whereby the hormone can bind to, and that is desensitization. And there is a solution for that, and we'll talk about it at the end of this lecture. So these are different factors. Okay. So let's talk about something else now. Hormones can be classified into different categories according to a number of criteria. That is their solubility, that is lipid soluble or water soluble, how they are transported, mechanism of transport is also important, and it depends on the solubility of the hormone. The chemistry of the hormone, that is the structure of the hormone itself. Is it, for example, a peptide hormone or a protein hormone, or is it a steroid, for example? How they are synthesized is also important. Processing and modification. Now, some hormones can be processed extensively and others may not. And we have a pharmacological way of classifying hormones. We will not talk about that, about the last one. So, let's talk about solubility. So, hormones can be classified according to their solubility. There are water-soluble hormones and we have lipid-soluble hormones. Water-soluble hormones include amino acids, proteins or peptides, as well as eicosanoids. The lipid-soluble hormones include steroids, thyroid hormones, as well as nitric oxide. Now, according to their solubility, transport is determined. So the idea is that the protein and peptide hormones, the water-soluble hormones, have no problem traveling by themselves in the bloodstream. But on the other hand, steroids, because they are lipid-soluble and as well as thyroid hormones, since they are less soluble in blood, they need a carrier, or let's say carriers. And most of them actually circulate in blood, complexed with other proteins like plasma globulins as well as albumins. And remember that albumin can bind to many, many molecules. So the free hormone, which is minimal in the blood, and bound hormone as well, they exist in equilibrium. And what's really important actually is the free hormone. This is the one that is free to get into the cell and affect a cell or make a cell respond in a certain way. So biochemical assays are important in considering not only the total hormone concentration, but also defining how much of that hormone is free. So it is really important to measure the free hormone, which is the active hormone, in blood. Except that measurement of the free hormone is technically not that easy. But keep that in mind. Now, binding of hormones to different molecules or different carriers can affect their half-life. So a free hormone, for example, would not have a long half-life. So catecholamines, for example, their half-life is like in the order of seconds. Well, consider the half-life of protein and peptide hormones, which is about they last for a few minutes. But for steroids and thyroid hormones, their half-lives are really long. They can last for hours or days even. The reason is that they are bound and protected from any enzymatic modification or clearance. So as I said, it's important to regulate the level of hormone that exists in the blood. And the level of hormone is dependent on a number of factors. Again, we have the rate of production, how much of that hormone is synthesized and secreted, rate of delivery, so we have the dilution factor, the proximity of the gland or the producing cell from the target cell, and the rate of degradation and clearance as well. So these are really important factors in determining how much hormone there is in the body. There are other factors that are important and they play a role. One of them is gender, for example, and that's why if you look at any blood test for hormones, there are indicated ranges for males, and these are different than those used for females. We have age as well, that can play a role in determining the level of hormone in the blood. Changes in the binding protein, the level of this binding protein, the developmental stage, especially in the embryonic stage, the reproductive status, whether a person is reproductively active or not, and the stage of the temporal rhythm as well for certain hormones. Okay. Now, hormones can be classified, as I said, according to different types, and one classification is basically whether they are lipid-soluble or not. Another classification is to what receptor they bind to. Is it an intracellular receptor, or is it a cell surface receptor? But there's another classification, and this classification is based on the chemistry of the hormones themselves. So we have different types of classifications. Hormones can be peptides, so we're talking about small molecules, peptide or proteins. So we're talking about small and large hormones like insulin, glucagon, etc. We have hormones that are actually amino acid derivatives, like the thyroxine, for example, the epinephrine, dopamine, serotonin, all of these neurotransmitters. We have steroids, which are derived from cholesterol, and they're modified, and we'll talk about that in the next lecture. We have eicosanoids, and these are derivatives of the unsaturated fatty acid, arachidonic acid. We talked about that in biochemistry. And we have gases as well, or an example is nitric oxide. So this is the chemical classification. So right here you can see the differences in structure of these hormones. These are the peptide and protein hormones, a sequence of amino acids connected to each other. We talked about that before. We have the steroid hormones that are all derived from the cholesterol nucleus of the cholesterol molecule. We have amino acid derivatives like the thyroid hormones, for example, and so on. So these are the eicosanoids, prostaglandins, thromboxanes, and so on, and so on. So now these hormones can be processed and modified, and some of them can be processed and modified, and some may not be processed and modified. And the location of processing and modification is really important as well. It can differentiate hormones from each other. So we have hormones that are synthesized and secreted in the final form, including aldosterone, estradiol, catecholamines, and so on. But we have hormones that are modified directly in the target cell. An example is the insulin, for example. That is the conversion of the pro-insulin, the active form of the hormone. And we have hormones that are modified indirectly by non-target tissues like thyroxine and testosterone. So we have testosterone is released from the source cells, and they get into other cells, non-target cells, where it can be converted to dihydrotestosterone. Dihydrotestosterone is then released, and it goes to target cells. Vitamin D3 as well. The conversion of vitamin D3 from skin to liver to kidney is an excellent example. Okay. Now, in terms of hormone synthesis, it's different according to the hormone type. So protein and peptide hormones can be synthesized differently than steroid hormones, amine hormones, and eicosanoids. The protein and the peptide hormones can also be synthesized by different mechanisms. We have alternative splicing, we have post-translational modification, and we have formation of synthesis, the early synthesis of a pre-pro hormone that needs to be activated into the final form. Okay, so let's talk about these first, the protein and peptide hormones. Now, one example, we have synthesis of peptide hormones themselves. Excellent examples include vasopressin and oxytocin. Now, these are 9 amino acid peptide hormones. They have really almost identical primary structure, amino acid composition and order, except for 2 positions. That is position 3, where you have isoleucine, and position 8, where you have leucine in oxytocin. In the vasopressin, you have phenylalanine and arginine instead. Otherwise, they're almost the same. These are the 2 important amino acids for the activity of these hormones. And they're produced by different genes. Now, we have certain hormones that are produced by alternative splicing. We talked about that in the molecular biology course. We have the calcitonin and calcitonin gene-related peptide. They're produced from the same gene, except that they are alternatively spliced at the RNA level. So right here you have the C1, C2 exons shared among the 2 hormones, but then we have different exons that exist in these 2 molecules, these 2 hormones. Okay. Now, an extreme example is hormones that are produced from the pro-opiomelanocortin peptide. So this is a large peptide that is produced from a single gene, and then it can be modified post-translationally. So after the formation of this polypeptide, it can be cleaved into different peptides, and these different peptides, each one of them has a certain action. So for example, adrenocorticothyroid hormone is important for the production of cortisol. You have the production of different enkephalins and endorphins, all from the same peptide. We have production of the melanocyte stimulating hormone as well. This is important for pigmentation of cells, of skin cells. In terms of finally synthesizing the pre-pro hormones, like insulin and glucagon, for example, they are produced on the surface of the endoplasmic reticulum. Once they are internalized into the ER, the pre-region is cleaved off. This is the signal sequence. It gets cleaved off, and you have production of the pro hormone. This pro hormone can then be cleaved or modified enzymatically into the active form of the hormone. So this is the third type of peptide and protein hormones. So here's an example again. The insulin itself, it's basically produced from a single gene, single polypeptide. You have formation of disulfide bonds, and then you have cleavage of the C peptide. That is, this portion of the peptide is cleaved off, forming the final form of this insulin molecule, having the A and the B chain. We talked about that as well, about insulin, in the molecular biology course, when we talked about cloning and how we can actually clone the different portions of the genes in different bacteria, and then we can combine the A peptide with the B peptide, forming the final mature form of the insulin. Now, this is just to show you, this table just shows you the differences in structure or amino acid composition of these different hormones. Notice that you have the thyrotropin releasing hormone. It's composed of just 3 amino acids relative to these, the vasopressin and the oxytocin, which are composed of 9 amino acids. And we have insulin, a large peptide molecule, large protein molecule relatively. So I don't want you, of course, to remember the numbers of amino acid composition, but I want you to know which one of them is large and which is small. Now, we'll talk about steroid hormones in the next lecture, but they're all synthesized from cholesterol in the mitochondria and smooth endoplasmic reticulum. They require different enzymes, and these enzymes are really important in sequentially producing these steroid hormones. These enzymes are expressed in different cells, and they're controlled by different hormones as well. The amine hormones, like the catecholamines, for example, they're produced from amino acids like tyrosine and tryptophan. We will talk in more details about these hormones in the CNS module. Eicosanoids, maybe we'll touch upon these at the end. These are all produced from arachidonic acid. You have then the production of the different eicosanoids, the prostaglandins, thromboxanes, and leukotrienes. Now, another classification, as I mentioned, is the pharmacological classification of hormones. You can have hormones that are agonists. You can have hormones that are antagonists, like insulin and glucagon, for example. You can agonists like glucagon and epinephrine. You can have hormones that can function as partial agonists or partial antagonists, as well as other hormones that can function as mixed agonists-antagonists. We will not talk about these in this module, but maybe you'll learn about them in other courses related to pharmacology. Now, hormones do not act separately or independently of each other or other molecules. Rather, they work as a team in regulating homeostasis of our body, and their interaction or the way they communicate with each other can be classified or can be categorized in different types. You can have hormones functioning synergistically. You can have hormones that are antagonists to each other. You have a permissive effect, or you have integrative effect. What do we mean by all of that? So a synergistic effect is that hormones tend to cause the same effect. That's a synergistic effect. So ADH and aldosterone, for example, they both have the same final effect, as well as the follicle-stimulating hormone and estrogen and their effect on ovaries for the development of the oocytes. So that's a synergistic effect. Antagonistic effect is when you have hormones that have different functions or opposite functions. So insulin and glucagon, for example. Insulin is produced, it promotes the synthesis of glycogen, for example, whereas glycogen accelerates the breakdown of glycogen into glucose. Okay, so we have the permissive effect. What we mean by permissive effect is that hormones you need a hormone that can prime, that can prepare the organ for another hormone. Or you can have hormones that can function together, inducing or amplifying the effect. So examples include epinephrine and the thyroid hormone. Now, epinephrine by itself can function or can act on fat cells to stimulate the breakdown of triglycerides. But it would have a much larger effect if you have production of the thyroid hormones, the T3 and T4 hormones. Now, you can have examples where you have hormones that stimulate the presentation of receptors on the cell surface. Or you can have a hormone that would induce the synthesis of of enzymes that are necessary for the production of other hormones. And then we have the integrative effect. In this case, they work complementary to each other, and they have the same end result, like the parathyroid hormone and the calcitriol hormone. They both increase the level of calcium in blood. Moving on, feedback or mechanisms of regulating the secretion and release of hormones is important, and usually this mechanism is by feedback, whereby the final hormone would control the release of the early hormone. So I'll give you a few examples here. We have a simple feedback mechanism right here. A good example is glucose and insulin. So the level of glucose actually can regulate how much insulin is released. A high level of glucose would induce the release of insulin so that cells can take up glucose. They can convert glucose or conjugate glucose molecules to glycogen, building larger glycogen molecules, and so on. So that's a simple mechanism. When the level of glucose falls down, Ramadan Kareem, when the level of glucose goes down, then the amount of insulin released from pancreatic cells would also go down as well. There is a more complex feedback mechanism for regulating the release and production of hormones. This is an example that I talked about earlier. So you have the hypothalamic hormones that are released. They affect the pituitary gland, which release trophic hormones, and these hormones can function on other peripheral glands that produce other hormones. Now, the hormones produced by the pituitary gland can feedback regulate the hormones that are produced by the hypothalamus. At the same time, the hormones produced by the peripheral glands can also feedback inhibit the release of hormones from the pituitary gland and the hormones released from the hypothalamic gland as well. Okay. Now, this is an important concept right here, which is the release of hormones is actually not continuous. Rather, it occurs in pulses. Okay? And this is really important. So, like luteinizing hormone, for example, you have production of this hormone, it peaks, and then it goes down. And then it peaks again, and it goes down in different time points. And this is really an important concept. This is an important feature of these hormones. And probably one reason for this pulsatile secretion of hormones is that to limit or to prevent desensitization and to reactivate the signaling pathways of these cells. So as I said before, when you have a continuous production of large amount of hormone, this hormone binds to the receptor is internalized and it gets degraded. So if you have continuous level of hormone in the blood, then there wouldn't be any receptor on the cell surface. But if the release occurs in pulses, so you have internalization, so you have hormone binding to the receptor is internalized, gets degraded, and when the hormone disappears, the receptor appears on the cell surface again. You have production of hormone binding to the receptor, and so on, and the cycle continues. So for most of these hormones, the secretion is pulsatile. Okay. There are other examples of such mechanism of release. Nerve impulses, for example, control the release of epinephrine. Blood calcium ion regulate the secretion of the parathyroid hormone, so it goes up and down. And the ACTH hormone from the anterior pituitary gland can stimulate the release of cortisol by the adrenal cortex. These are all examples of pulsatile secretion of hormones. So this is the end of this lecture. Hope you enjoyed it. I know it's a lot. Take it easy. Enjoy listening to it. And I'll see you next lecture.